Welcome everyone to the lecture on forces of evolutionary change. Our objectives with this lecture are to demonstrate an understanding of how population genetics and evolution relate to each other. For population genetics, you should be able to define terms common to population genetics, list 3 methods used to distinguish species from one another, list factors affecting genetic diversity in the populations of species, and describe how Hardy-Weinberg concept is applicable. For evolution and natural selection, you should be able to describe the development of evolutionary thought from historical perspectives, describe how the concepts of evolution and adaptation are related, explain how natural selection works, describe the mechanisms of evolution other than natural selection, and describe how human evolution is supported by scientific evidence. And actually, human evolution we're going to talk about more in lab, so I won't focus very much on that here. All right, evolution is genetic change over time. It occurs when there are changes in heritable traits from generation to generation. Some changes can lead to the development of new species. Now, evolution occurs in populations, not individuals. You are born with the same DNA that you die with. Your individual DNA or alleles don't change, but the allele frequencies in populations do change over time. Some alleles become more common, others become less common. That's evolution. For example, allele frequencies vary between populations. Like in the population of Asia, the frequency of alleles that produce black hair is high. The allele frequency is calculated using the following equation. So the number of copies of the allele for black hair, say, divided by the total number of alleles for the gene for hair color in the population. Evolution is detectable by examining gene pools. In the population of Sweden, the frequency for alleles that produce black hair is low, and the gene pool for a population in Sweden differs from that of the gene pool or the population in Asia. If Swedish people migrate to Asia and interbreed with the local people there, then allele frequencies in Asia will change, right? And evolution will have occurred. So take a moment, check your understanding. Which of the following can evolve? A group of fir trees in Oregon, Earth's climate, a single fly as it develops, a fossilized turtle, or a cell in your body whose DNA develops mutation. Good. So evolution occurs in populations. A group of trees in Oregon is a population of fir trees, right? All right, take a moment and explain to yourself, how do allele frequencies for each gene determine the characteristics of a population? All right, let's talk a little bit about the history of evolutionary theory. So here's a brief timeline. If we were to go back all the way to Aristotle, 350 BCE, individuals in a species are basically identical and species are unchanging. Fast forward 2,000 years to Buffon. Buffon is the one who stated that species change as they spread from their original location. So this is where the idea of a common ancestor and changing species first began to arise. In 1809, Lamarck stated that new species come from existing ones through environmental forces. And Lyell said all changes in nature are gradual. And this is where we kind of begin to come up with this basis of a testable definition for evolution. Charles Darwin and Alfred Russel Wallace are the ones who stated individuals in a population are different and species arise through the process of natural selection. So this proposed a mechanism for how it is that evolution leads to the formation of new species. Now, fossil discoveries began to reveal the history of life on Earth. Geologists have discovered that different rock layers reveal different groups of fossilized organisms, many of them now extinct. So older layers of rock contain older fossils, right? The further down you go, the older the rock, the older the fossils. The closer to the surface, the newer the rock, the younger the fossils. All right, Darwin's voyage. So Darwin took this famous trip around the world, leaving from London. He sailed around the coast of South America, visited the Galapagos on his way over to Australia, around the southern cape of Africa, back to South America before heading back to Europe. So while on this voyage, Charles Darwin documented the great variety of organisms in South America and their relationships to fossils and geology. And he began to think that these were clues to how new species originate or get started. Darwin saw that individuals and populations are different from each other. They're not all the same. And he read an essay by Thomas Malthus about how food availability, disease, and war limit human population size. And he reasoned that in any population, not just humans, individuals that can't obtain essential resources would die. Darwin proposed the idea of descent with modification. So Charles Darwin studied finches in the Galapagos Islands here, and he saw that their beak types matched up with the foods they eat. So finches that had really thick beaks ate really hard seeds, and finches that ate tiny little insects had skinny little beaks that were able to reach in and nip them off of plants. So their beaks matched up with their foods. Darwin thought that the different finch species likely all descended from the same finch ancestor. So if you look, these Galapagos Islands are way far off the coast of Ecuador. So the nearest land mass is super far away. And the Galapagos Islands is a small archipelago of islands. So there's more than one island. And on each of the islands, there's more than one species of finch. So the most likely explanation is that a single finch population migrated out, and it could have been as small as 2 birds, right? Or one fertile female migrated from the mainland out to the Galapagos Islands. The finch's population would have exploded and taken off, and then later diversified or undergone speciation on the Galapagos Islands. This is much more likely than the idea that 12 different migration events happened from 12 different species of finches on the mainland. And by the way, there are no species of finches on the mainland of Ecuador that are exactly the same. There are no species that exist on the mainland and also exist on the Galapagos Islands. All the Galapagos finches are unique relative to anything you'd find on the mainland. All right, so this is when Darwin first proposed the idea of natural selection. Charles Darwin saw that the environment on each island influenced the survival and reproduction of the finches living there. And finches with features better suited to the environment were able to survive and reproduce better than others. This is where that idea of survival of the fittest comes out. All right, artificial selection is also changes in allele frequencies. However, artificial selection happens when humans are controlling the breeding. So it's like natural selection, but humans control which individuals have reproductive success and which do not. All right, An example of this would be the wild mustard plant. Have you ever seen garlic mustard? It's an invasive species, but we have it here in Michigan. And it's related to this wild mustard. But anyway, plant breeders bred this mustard plant with traits that they liked, and it led to a variety of different plants being produced. So all these plants, if you're a gardener, belong in the group called Brassicaceae. Cabbages, kohlrabi, and kale, broccoli, Brussels sprouts, cauliflower are all very closely related plants. And they were created by selectively breeding or artificially selecting for specific traits from the wild mustard plant. Humans artificially alter allele frequencies all the time. We do it with dogs. Specifically, we choose to breed dogs with the traits we like together, and eventually those develop into specific dog breeds. However, when you focus to breed for specific traits, often what happens is we develop a byproduct where problems are also more common in those breeds because we've been hyper-focused on certain traits. So in artificial selection, a human chooses desired features and then only allows the individuals that best express those qualities to reproduce. This increases the frequency of desired alleles in a population. And as you can see, it also increases alleles for undesirable traits in populations sometimes too. All right, so Darwin proposed that natural selection is a mechanism for evolution. It's a thing that can make evolution happen. It changes allele frequencies from one generation to the next. So here's kind of the thought process behind that. First, we start with observations. So Darwin observed heritable or genetic variation within a species, and no two individuals are exactly alike. Some of this variation is heritable. It can be passed from one generation to the next. Another observation is that there are limited resources. So every habitat contains limited supplies of the resources required for survival. And 3, an overproduction of offspring. More individuals are born than what survive to reach reproductive age and actually successfully reproduce. Some inferences we can make from these observations are that there's a struggle for existence. Individuals compete for the limited resources that enable them to survive. There's also unequal reproductive success, and this is the natural selection part. The inherited characteristics of some individuals make them, more likely to obtain resources, survive, and reproduce. Descent with modification is another inference we can make here. Over many generations, a population's characteristics can change by natural selection, even giving rise to new species. All right. Darwin and Wallace laid the foundation for evolutionary theory. Both scientists independently came up with the idea of natural selection after years of studying nature. So if we go back in time, we see Darwin published his On the Origin of Species. This is his book where he first put forth the idea of evolution by natural selection. Now, Darwin came up with the same idea around the same time. Darwin did not know about Mendel and the basic laws of inheritance. They were contemporaries. They lived and worked at the same time, but they didn't know about each other's work until much later. So these two theories were developed independently, but they fit together nicely. All right, so Mendel's laws were continued to work on. Some other scientists were able to map genes onto chromosomes. We also have decay rates of isotopes reveal that the Earth is billions of years old. So up until early 1900s, we didn't really have any solid, quantifiable evidence about how old the Earth really was. And this fits nicely with Darwin's On the Origin of Species. Evolution takes a long time, sometimes, and sometimes it can happen quickly, but generally speaking, evolutionary change is usually gradual and slow. All right, and then in the 30s and 40s, modern evolutionary synthesis or theory unifies the research on genetics and evolution. This is when we're able to put these two ideas together. In the 50s, Watson and Crick and Rosalind Franklin discovered the structure of DNA. In the 60s, the theory of plate tectonics explains continental drift. It also explains why we see certain organisms distributed across the continents the way we do. In 1977, a scientist named Weiss reclassified life into 3 domains. Eukarya, which is plants, animals, protists, and fungi. Archaea, these are single-celled extremophile organisms, and domain bacteria. In the 90s, large-scale genome sequencing began, reinforcing genetic similarities among species. And even now, there's still ongoing research about gene function and regulation to reveal the origin of new phenotypes. Evolutionary theory is supported by data. Excuse me. And evolutionary theory continues to expand. So modern evolutionary synthesis includes what scientists have learned since Darwin's time, particularly about genetics. Heritable traits are passed in our DNA. Genetic mutations and sexual reproductions are continuous sources of heritable variation. And genetic variation is the raw material that evolution acts on. If an allele is fixed in a population and all the individuals have the same 2 alleles, can natural selection select for an individual that's more successful because it has one trait over another? Well, no, because all the individuals in the population would have the same phenotype, the same trait. So we have to have genetic variation in order for evolution to occur. All right, evolutionary theory is also often misunderstood or misrepresented. So some common misconceptions are that evolution's just a theory, so it might not be true. Well, in science, the word theory means that it's an explanation that's been tested and unites many observations. Evolution has been tested countless times, and evidence for it is strong. Gravity is also a theory, right? No one doubts gravity, and no scientists doubt evolution. All right, biological evolution explains the origin of life. This actually isn't true. Evolution doesn't begin until after life already exists. Another misconception is that intelligent design is an alternative scientific theory that explains the diversity of life. Well, intelligent design is not a scientific theory because it can't be tested. No future observations can disprove the idea that an intelligent creator designed life on Earth. Intelligent design is a faith-based argument, not a scientific one. Another misconception is that evolution is random. While some mechanisms of evolution, like mutation, do occur randomly, natural selection, however, is non-random because the environment selects against poorly adapted individuals. And one more misconception I'll address here is that in a changing environment, all individuals in a population develop beneficial adaptations at the same time. And this is false. Adaptations can become fixed in a population over many generations, as individuals with beneficial adaptations are more likely to survive and reproduce and pass on their genes. But it doesn't happen instantly. Remember, individuals can't change their DNA. All right, so let's do a comprehension check here. Which of the following statements supports the concept of natural selection? Individuals with traits best suited to the prevailing conditions tend to leave more surviving and fertile offspring. Traits that increase the survival and reproduction in the current generation will be more common in the next generation. A and B, or none. The best answer is A and B are both correct. All right. So what did Darwin observe that led him to develop his ideas about the origin of new species? Explain his logic. Write down an explanation to address this question in your own words. How are species differentiated? Well, we have 3 different ideas that biologists are still kind of arguing over which is right. I'll explain all 3 of them here. So the morphological species concept is basically that we can group organisms based on their physical similarities. This is the way that species were grouped and described prior to the development of genetic technology and phylogenetic trees or species trees. It's still useful and used, especially when we're looking at extinct organisms or things that are really difficult to find and study, like giant squid. We have very few giant squid specimens to look at, right? Or deep sea creatures. We just don't have a lot of data on them. So using the physical characteristics of one good sample can be very useful to give us a clue of where they fit into the tree of life. A more commonly applied definition of species is the biological species concept. And it's this idea that if there are 2 populations that can interbreed in nature and they produce fertile offspring, so their offspring can have babies of their own, then those 2 populations are populations of the same species. It's a very practical explanation. So an example of this would be tigers and lions can mate. They can hybridize and produce a liger or a tigon. But these hybrid cats are not fertile. They can't have babies of their own. And tigers and lions live on separate continents. They wouldn't breed together naturally. So tigers and lions, even though they can interbreed, are separate species. All right, and then a third way that we have to define species is based on their genetic data. So we call this the lineage species concept. We can sample individuals and sequence their genomes, then apply a statistical model to tell us the probability that they come from one species or more than one species. And this has been a really influential tool as genetic data has become more available, and especially sequence data for whole genomes since probably around the 2010s is when it began to become common for whole genomes to be sequenced. And it gives us a wealth of genetic data. Now, this is something that's kind of, I'm trying to think of what's the right word, not hotly debated, but it's a hot topic in biology, is where do we draw the line between what are 2 different populations and 2 different species using genetic data. And there are some problems with it. For example, some species have more genetic variation within a single species than other groups of organisms do between higher levels of classification. So, for example, the split-gill mushroom has 20% nucleotide variation within its species. So, for every 100 A's, T's, G's, and C's in the genetic code, 20 will be different just between 2 different individuals of the same species of mushroom. By contrast, humans have 0.1% nucleotide variation. So that would be out of every 1,000 letters in your genetic code, there would be 1 difference between you and any other human on the planet. So it's difficult to say that if there's, 25% nucleotide variation, then those are different species, because that would lump humans in with things like probably monkeys with tails. So it's difficult to figure out where those lines are. And in reality, the best way to say two groups of organisms are different species is to use a combination of all three species concepts. There should be physical differences, but not always. There should be affective differences in how they function in nature, but not always. And there should also be a genetic basis for having different independent gene pools on the level of species differences. So what exactly a species is something that biologists are still working to define. There's not a clear-cut answer, which I think is pretty fascinating, given that we think of it as being something very basic. All right, so natural selection molds evolution. Natural selection does not create alleles. Instead, it can strongly select for alleles that have already arisen by chance. There has to be genetic variation. There has to be more than one allele for a trait in a population in order for natural selection to have any effect. For example, this seahorse's alleles produce a phenotype that allows it to blend in with its surroundings, which is useful for evading predators. Natural selection requires genetic variation. Since more individuals are born than the resources in an environment can support, the struggle to survive is inevitable. Some individuals in a population are better than others at surviving and reproducing. Adaptations enhance reproductive success. So reproductive success or fitness is the number of babies that you get to put into the next generation, right? So my own reproductive success is about to go up. My own fitness is about to go up because I'm about to have another baby, right? All right, adaptations are heritable features that improve an organism's ability to survive and reproduce. For example, some people produce more gametes than others. Short-term changes in an individual are not evolution. Remember, evolution has to do with change in your allele frequencies, and individuals don't change their DNA. Populations do, but not individuals. All right, a good example of this that happens quickly is antibiotic resistance in bacteria. So say we have a cut that becomes infected. There's bacteria that are just regular, normal bacteria. A mutation can occur that produces a new allele. And let's say as that cut becomes infected, as the population of bacteria grows, you go to the doctor and you get a prescription for an antibiotic. Well, that antibiotic can kill all of the green bacteria because they're not resistant. But this little red mutant bacteria here happens to carry an allele that allows it to be antibiotic resistant. And now, after taking antibiotics, the cells that are left, or the bacteria that are left in your cut, are all the ones that are resistant to that antibiotic. And this is, in short, how antibiotic resistance evolves. Bacteria that are resistant to antibiotics have an adaptive advantage that the non-resistant bacteria lack. When antibiotics are present, those resistant bacteria can survive and reproduce better than the others. So there's no benefit to being antibiotic resistant here. But once we get here, when the antibiotic has been released in the environment or in your body, now there's a big advantage to having that mutation that allows you to be resistant to the antibiotic, or allows the bacteria to be resistant. Adaptive alleles become more frequent in the population over time. The antibiotics don't create a resistance allele. The variations for resistance was already present in the population. It just allows that one to be beneficial. Natural selection eliminates poorly adapted phenotypes. So as environmental conditions change, the phenotypes that natural selection favors will also change. And adaptations that seem perfect in one environment could be completely wrong in another. Natural selection can even eliminate entire species. If beneficial alleles aren't available at the right time, an environmental change can wipe out a whole species. And the species shown below have become extinct. So this is a creature, I think, from the Devonian period, so about 350 million years ago, called a Eurypterid. It's like a crustacean. And then these are logs from the Petrified National Forest. All right, another thing to keep in mind is that evolution doesn't have a goal. So here's a picture of an orchid, and this wasp is its pollinator. They've co-evolved. They've evolved alongside each other. But the orchid doesn't evolve in order to be better pollinated by the wasp. The wasps created a selective advantage for orchids that happen to have the right shape for wasps. All right, fitness is reproductive success. So by itself, survival is not enough for evolutionary fitness. Fitness depends on the ability to reproduce, or for the organism's genes to be part of the next generation. The more of your genes that you get into the next generation, the higher your fitness is. So here's a quick comprehension check for you. Many plants require moisture to reproduce. What will happen to a plant population during a prolonged drought? Well, some plants acquire the ability to reproduce without water. If none of the plants can reproduce without water, the whole population might go extinct. Or those plants that can already reproduce without water will pass on their alleles to the next generation, and others will not. Or are both B and C correct? And both B and C are correct. So if there are no alleles in the population already for being able to reproduce without water, then they'll all go extinct. If there are alleles in the population for being able to reproduce without water, then those individuals will have an advantage and be able to reproduce and become more common in the next generation. Good. All right. How can natural selection favor different phenotypes at different times? I want you to jot down your own thoughts to this question in your own words. All right? All right. Evolution is not only possible, it's unavoidable. In real populations, allele frequencies change over time because they're affected by so many selective forces. And we can test evolution. So scientists test evolution and measure whether allele frequencies do or do not change from one generation to the next. In this hypothetical population of ferrets, allele big D produces dark fur and allele little d produces light fur. Counting the alleles allows us to calculate their frequencies. So you can look at this picture and count the number of big D's and little d's. We can use these allele frequencies in an equilibrium equation to determine if evolution is happening or not. And the name of that equilibrium equation is called Hardy-Weinberg equilibrium. Hardy-Weinberg equilibrium is a hypothetical situation in which allele frequencies do not change between generations. If there's no change in allele frequencies over time, then that means that evolution is not occurring, right? It can be compared to real populations to see if real populations are changing in their allele frequencies over time, which can be very useful. All right. So if allele frequencies stay the same, then that means that there is random mating, there is no migration, there's no genetic drift, mutation, or natural selection occurring in that population at that allele. The allele frequencies stay the same over time. Factors that can alter allele frequencies, or things that cause evolution, could be if there are some individuals with low reproductive success due to environmental change. Hardy-Weinberg equilibrium relies on assumptions about a population. And a population meeting all the assumptions of Hardy-Weinberg is said to be at Hardy-Weinberg equilibrium. And these are the assumptions: random mating, no migration, no genetic drift, no mutation, and no natural selection. If this is true, then the population is not evolving, and allele frequencies at the beginning should be the same as allele frequency at the end of whatever time frame you're looking at. We can actually calculate allele and genotype frequencies. If the assumptions of Hardy-Weinberg equilibrium are met, two equations represent the relationship between allele frequencies and genotype frequencies. So if we look, here is the equation for allele frequencies. And all this means is that if there are two different alleles for a gene, allele P and allele Q, they have to sum, their frequencies have to sum to 1, right? So basically, if we're going to break something down by percents, there has to be 100% when they're all done. P is the frequency of the dominant allele. So in our ferret example, that would be the frequency of the uppercase D allele, which you can look at that picture and calculate, but it's 60% or 0.6. Q is the frequency of the recessive allele, or in this example, the lowercase d, for lighter colored fur, and that's 40% or 0.4. P plus Q have to equal 1. 0.6 plus 0.4 equals 1. So it all checks out. Genotype frequencies, now these go back to a Punnett square, like we did last week. We've got Punnett squares, so here's homozygous dominant, big D, or P squared. And you can calculate that, right? P squared would be P times P, or 0.6 times 0.6. Means 36% of the time we should have homozygous dominant dark brown ferrets. The next part of this equation is plus 2pq. This is the total number of heterozygotes, or the frequency of heterozygotes. 2, because there's 2 boxes, right? Times the frequency of P and Q. So 0.6 times 0.4 is 0.24, times 2 should be 0.48. There we go. And then adding onto that, the frequency of the homozygous recessive individuals would be these light-colored ferrets. So 0.4 times 0.4 equals 0.16. So we can make predictions about the frequencies of genotypes and phenotypes based on the frequencies of alleles in populations. I won't ask you to do these calculations, but I will ask you to understand the assumptions of Hardy-Weinberg. I will ask you to understand how allele frequencies and phenotypes are related. And I will expect that you understand how Hardy-Weinberg can be used as a test to identify populations that are undergoing evolution and those that are not. All right, so in real populations, the assumptions of Hardy-Weinberg are always violated, meaning evolution is always taking place. Natural selection and mutations do occur. There are random changes in allele frequencies, and it's actually pretty common. Some individuals rarely mate at random. There's usually some type of mate choice. And there's always some level of migration between populations. All right, so Here's a comprehension check question for you. A population of 100 starfish is in Hardy-Weinberg equilibrium. The trait for long arms is completely dominant to the trait for short arms. And in this population, 40% of all alleles for this trait are recessive. 60% of all alleles for this trait are dominant. How many individuals would you expect to be homozygous dominant? Well, if you go back to that second Hardy-Weinberg equation, p squared plus 2pq plus q squared equals 1. P, remember, is the frequency of the dominant allele. And Q is the frequency of the recessive allele. So we know P equals 0.6, right? That's the decimal that goes with 60%. And if we want to know how many individuals are, we have to square this. So 0.6 times 0.6 equals 0.36. And since our population size is 100, that means that there's 36 individuals in the population that should be homozygous for the dominant allele. All right, if you were studying a population over time, how could you measure whether or not evolution occurs? Take a moment, jot down your thoughts here in your own words. All right, so there are several different ways natural selection can shape populations. 3 different types of natural selection have effects on the phenotypes in a population. There's directional selection, where selective force or change in the habitat forces the frequency of alleles to shift. So either a movement to the left or the right, where before most of the moths were medium color, but after this selective force, most of the moths are darker in color, right? There's also disruptive selection. Something changes, and having that common intermediate phenotype is disadvantageous, or it's bad. Those snails are more likely to be eaten. Then we see a shift towards light-colored and dark-colored snails. And then another type of natural selection is stabilizing selection. So there's actually really strong natural selection for the size of newborn babies. If babies are too small, they have lower survival rates. And if babies are too big, they also have lower survival rates, which is why we generally see a very narrow range of birth weights for full-term human babies. All right, why are harmful alleles sometimes maintained in a population? Well, one explanation for why harmful alleles persist is a heterozygote advantage. And this is where heterozygote is favored over homozygotes. And a really good example of this harmful recessive allele is sickle cell disease. So normal red blood cells have this nice round shape, but people who are, so if you are heterozygous for the sickle cell allele, you will have normal shaped red blood cells. If you happen to have 2 copies, if you're homozygous recessive for the sickle cell allele, then you will have sickle cell disease, where your blood cells are shaped kind of like this crescent moon shape, and it causes a lot of problems because these will become plugged up or blocked up in your blood vessels. It can cause pain, anemia. It can be a real problem for your health. However, people who are heterozygous are resistant to malaria. Malaria is an infectious disease spread by mosquitoes, and in parts of the world where malaria is common, sickle cell alleles are more frequent in the population than in malaria-free parts of the world. So there's a benefit to being heterozygous for the sickle cell allele if you live in a part of the world where malaria is common. So heterozygotes for the sickle cell allele do not have sickle cell disease, and they're protected against malaria. If two heterozygotes mate, their child has a 25% chance of inheriting sickle cell disease. It also has a 50% chance of not having sickle cell disease and being resistant to malaria. All right, let's do this one. So in very sunny places such as Africa and India, exposure to UV radiation selects for darker skin color. What type of natural selection does this example illustrate? Stabilizing, disruptive, or directional selection? Very good, directional selection. All right, explain how habitat change can lead to directional, disruptive, or stabilizing selection. Jot down your own thoughts here in your own words, please. All right, sexual selection. This is natural selection, but specifically related to mate selection and reproductive success. So directly influences reproductive success. These are behaviors like building complex nests, flashing showy plumage, butting heads with rival males, all things that appear to waste energy. Sexual selection is a force that can favor traits that apparently reduce survival, right? Like picture a bright red cardinal. They perch out on the top of a tree or way out on the tip of a tree branch. They're bright red and easy to see, and then they start singing. And it makes them super easy for a predator to find and catch, right? But being bright red and singing loudly are all factors that attract a mate and make it more likely that they'll actually have reproductive success. Sexual selection is a type of natural selection. It results from variation in the ability to obtain mates. This can include competition for access to the other sex, or from one sex choosing attractive mates of the other sex. Sexual selection causes evolution to occur because individuals with these sexually selected traits mate the most and therefore pass along their genes the most. Mating is not random when sexual selection is acting. And since Hardy-Weinberg doesn't apply, evolution occurs. For example, male birds of paradise have evolved bright plumes to attract females. All right, how does sexual selection promote traits that could decrease survival? Write down your thoughts here in your own words, please. All right, mutations cause evolution to occur. So mutations create new genetic diversity. Beneficial mutations are passed on to the next generation, so their frequency can increase. Genetic drift is another evolutionary force, and it's basically sampling error. Sometimes, just by random, only some individuals in a population get to reproduce, causing a change in allele frequency to occur purely by chance. Drift is strongest when population sizes are small. But this is a nice little visual of it here. So if this jar of marbles is representative of all the alleles in a gene pool, or all the gametes, a random sample, so if you were to pull out 10 just by chance, even though there's an equal number of red and blue alleles or marbles in this jar, you might pull out 7 red ones and 3 blues. And then from there, you'd stock another jar with that allele frequency, 70 reds, 30 blues, and then you sample again. This time, say you might end up with 9 reds and 1 blue. Restock the jar again, and then you can see after just 1, 2, 3, by the time we get to the 4th generation, the allele frequency has changed entirely, right? And the blue allele has been lost, the red allele has become fixed. And from this point on, unless there's a mutation or a migrant individual with different alleles, all the individuals will have the red allele for this trait. So genetic drift can cause evolution to occur. Unlike mutation, which increases genetic diversity, Drift tends to eliminate alleles from populations. And again, the smaller the population is, the greater the impact of genetic drift. All right, the founder effect is another example of genetic drift. So when just a few individuals migrate away to establish a new population, the allele frequency might change. So if this is the genetic diversity in an original population, when just a few individuals start a new isolated colony, they'll only bring a sampling of all those genetic alleles to the new population, right? And that's called a founder effect. We see it a lot on islands. All right, population bottlenecks can also cause genetic drift. A population bottleneck occurs if the size of a population becomes greatly reduced. Genetic diversity in the new population decreases as many of the alleles are lost. Only the alleles in the surviving individuals will be maintained. So if you think of something like cheetahs, where they were hunted to near extinction, cheetahs underwent a dramatic population bottleneck. There used to be lots of cheetahs, lots of genetic variation. They were hunted so extremely that there are very few cheetahs left in the wild. So now we have much less genetic diversity in the remaining cheetahs that are left. And actually, cheetahs today are so inbred, they're so closely related to one another. If you take a skin graft off any one cheetah and put it on another cheetah, it will accept it as its own tissue because there's so little genetic variation between the remaining cheetahs we have. All right, non-random mating also causes evolution to occur. So we talked about sexual selection or mate choice and artificial selection. These factors tend to concentrate alleles locally, which affects allele frequencies. Many other factors also influence mating, including geographic restrictions and physical access to the opposite sex. So it could be something like a freeway could be a pretty solid barrier to gene flow for something like a frog, right? All right, gene flow causes evolution to occur. And gene flow is how alleles move between populations and increases genetic variation. This can change the allele frequencies in both populations. So say we have two populations of rabbits. If an individual from the light-colored rabbits migrates into the dark-colored rabbit population and interbreeds, that will change the allele frequencies here, right? All right, so keep that in mind. Evolution, what it boils down to, is a change in allele frequencies over time. Each mechanism of evolution changes allele frequencies. And the conditions for Hardy-Weinberg equilibrium don't normally occur together in natural populations, but it's useful to compare natural populations to this Hardy-Weinberg kind of standard to see how things are changing. All right, mastering concepts. So South China tigers have two color patterns, orange and black or blue and gray, which don't affect an individual's reproductive success. Over many decades, the tiger population has drastically declined, and all the blue-gray individuals have disappeared. What evolutionary process eliminated this color pattern? All right, and one more good example here. So bacterial evolution goes hog wild on a farm. Prior to last summer, or the summer of 2023, it was common for antibiotics to be added to livestock feed in order to improve growth rates. Not to keep the animals healthy, but a side effect of these antibiotics is an improved growth rate. So farms where animals like hogs were continuously fed antibiotics have actually become breeding grounds for antibiotic-resistant bacteria. So we see here, if we compare the number of resistance genes detected in the soil on farms that feed their livestock antibiotics regularly relative to the number of genes for antibiotic resistance that we just find in the soil in the same area but not on those farms, there's much higher, like double the amount of genes for antibiotic resistance. And this is actually why the FDA instituted the veterinary feed directive in the summer of 2023. So it's antibiotics can only be given to food animals now with a veterinary prescription and veterinary oversight, because this is a huge problem for public health. We need the few antibiotics that we have to be effective for human health. We can't have all these resistance genes being selected for in our food supply. It's too risky. So this is a way that evolution can impact your daily life. It affects what you eat. All right, so by now, you should be able to demonstrate an understanding of how population genetics and evolution relate to each other. You should be able to use common terms in population genetics. List 3 methods to distinguish species from one another. List factors affecting genetic diversity in populations. And describe how the Hardy-Weinberg concept is applicable. For evolution and natural selection, you should be able to describe the development of evolutionary thought from historical perspectives. Describe how the concepts of evolution and adaptation are related. Explain how natural selection works. Describe the mechanisms of evolution other than natural selection. And we'll address number 5 in lab and talk about our own evolutionary histories in the lab. All right. As always, any questions you have, please write them down. Bring them to student office hours or class, or ask a classmate. But make sure that you keep track of your questions and find some answers. All right.